Hello students, last lecture we studied about proteins. Now in this lecture we'll be doing enzymes. Now what are enzymes? Enzymes are also naturally occurring biomolecules. The word enzyme was derived from Greek word in leaven or in yeast to describe this process. So what is enzyme? What is the definition of enzyme? Enzymes are the naturally occurring biocatalysts. They are produced by living cells and which catalyze many biological reactions in plants and animals. So that is, they are the biomolecules which act as a catalyst. What are catalysts? Catalysts are the substances which increase the rate of reaction. So basically, they catalyze the reaction, they increase the rate of the reaction. And which reactions? Biological reactions that happen in plants and animals. Or the other definition is the proteins which perform the role of biological catalysts in the body are called enzymes. So every living cell contains at least 1,000 different enzymes, and most enzymes catalyze only one reaction or one group of similar reactions. Thus, enzyme catalysts are specific, highly specific in nature. For example, insulin. Insulin is an enzyme secreted by pancreas and it controls the blood sugar level. Amylase. Amylase is an enzyme present in saliva that hydrolyzes starch into glucose. Then maltase. Maltase is the enzyme which hydrolyzes maltose into glucose. Then lactase. Lactase is an enzyme which hydrolyzes lactose into galactose and glucose. Then zymase. Zymase is an enzyme that catalyzes the fermentation of sugar. And sugar after fermentation forms ethanol and carbon dioxide. Now, what is the mechanism of enzymatic reactions? What is the mechanism of enzyme? So, how enzyme works as biocatalyst? So, there are three steps. In step 1, formation of enzyme substrate complex takes place. So, enzyme combines with the substrate and enzyme substrate complex is formed. So, this is an enzyme. It is having active site here. When the substrate forms bond, enzyme substrate complex is formed. This is the substrate. So enzyme and substrate, they react, they form bond and enzyme substrate complex is formed. Then in the step 2, what happens? Formation of enzyme product complex takes place. So enzyme substrate reacts with product and enzyme product complex is formed. How? So here on the active side to which substrate is attached, the reaction of substrate takes place, product is formed and a new bond between the enzyme and the product is formed. So basically enzyme product complex is formed. Now in the third step what happens? Let's see. In the step 3, separation of enzyme and products takes place. So enzyme product complex gets separated into enzyme and product. So enzyme and products are released. So these products are released, enzyme remains and waiting for the another substrate to form bond. So this is how in 3 steps the mechanism takes place. So first this is the substrate, this is the active site on the enzyme. Substrate and enzyme bond, enzyme substrate complex is formed. Then substrate reacts to form products and products get detached from enzyme and enzyme is ready for another substrate to form bond. So formation of the enzyme substrate complex has a very low activation energy. That is how the rate of the reaction is very high. So some enzymes are so efficient that one enzyme molecule can catalyze the reaction of 10,000 substrate molecules in 1 second. So basically it is having a low activation energy. So enzyme substrate complex has got a very low activation energy. That is how the rate of the reaction is very high. So basically with the help of the enzyme or the catalyst, reaction follows a different path. That means a path of the low activation energy. So activation energy is energy required by the molecules to form products. So when the activation energy is low, the product formation will be fast. Therefore, the rate of the reaction is very high. Then industrial application of enzyme. First application is conversion of glucose to fructose. So for this, the enzyme used is glucose isomerase. Second application is manufacture of new antibiotics using penicillin G-acylase enzyme is used. Then manufacture of the laundry detergents. The enzyme used is proteases. Manufacture of the esters used in cosmetics using genetically engineered enzyme. So basically all these enzymes are catalyzing the reactions. So these are some of the questions based on enzyme. Define enzyme, write example of enzyme, explain the mechanism of enzyme, write application of enzyme. Next topic is nucleic acids. You all know that our body is made up of simple units called cell. Each cell contains nucleus. In the nucleus, the chromosomes are present, and on the chromosomes is present the genetic material DNA and RNA. So this DNA is a genetic material. So you can see here in the cell, DNA and RNA are present. So basically DNA and RNA are the nucleic acids. DNA is deoxyribonucleic acid and the RNA is ribonucleic acid. So what are nucleic acids? Nucleic acids are also the naturally occurring biomolecules. Nucleic acids are the principal genetic materials of all living organisms. The term nucleic acid is the overall name for DNA and RNA, the members of the family of biopolymers. And these nucleic acids, they contain carbon, hydrogen, oxygen, nitrogen, and phosphorus elements. What is the definition of nucleic acids? Basically, nucleic acids are also the biopolymers. They are found in nuclei of the living cells in the form of nucleosomes or nucleoprotein. Or nucleic acids are the biopolymers of the repeating units nucleotide. So if nucleic acid is a polymer, the monomer is nucleotide. Now what is nucleotide? Just shortly tell you. Let us see how the nucleic acid looks like represented. It is represented by it is basically the monomer of the nucleotide, and nucleotide is made up of three molecules. One is phosphate, sugar and base. Phosphate, sugar and base. So the unit nucleotide is made up of phosphate, sugar and nitrogenous base. These repeating units of the nucleotides forms the nucleic acids. The nucleic acids are of two types, DNA and RNA. So DNA is what? Deoxyribose nucleic acid, and RNA is ribose nucleic acid. So this DNA is actually made up of double-stranded helical structures. So that this helical structure, when you twist it, double helical nucleus structure. So this is helix is formed. So this twisted helix is two helix twisted together are called is the basic structure of DNA, and this RNA is a single stranded structure. How is a single stranded structure? You see, it contains only one helix. Okay, this helix contains sugar and phosphate molecules, and in between you see there are nitrogenous bases. So these nitrogenous bases in DNA are adenine, guanine, cytosine and thymine. while the nitrogenous bases present in RNA are adenine, guanine, cytosine and uracil. So this helix is made up of what? Sugar and phosphate. And the base unit attached to sugar and phosphate comes in the middle of the strands of DNA. So which nitrogenous bases are present in DNA? Adenine, guanine, cytosine and thymine. And which nitrogenous bases are present in RNA? Adenine, guanine, cytosine and uracil. So the first three are same, only one is different. That is, in DNA there is thymine, while in RNA there is uracil. Now this DNA is present in nucleus of the cell. RNA is present in the cytoplasm of the cell. This DNA has got a unique property of replication, while RNA hasn't got such a property. What is replication? That means the two helix of the DNA, they unwind and each helix can lead to the another DNA. So they unwind and each helix can form another DNA. So this property is called replication. This is shown by DNA and not by RNA. Then the sugar present in DNA is 2-deoxyribose sugar, while the sugar present in ribonucleic acid RNA is ribose sugar. So this contains our DNA contains bases adenine, guanine, thymine and cytosine, while RNA contains adenine, guanine, uracil and cytosine. So DNA is stable and does not hydrolyze easily by alkalis, while RNA is unstable and gets easily. So nucleotides. Nucleic acids are the unbranched polymers of repeating monomers called nucleotides. DNA contains several million nucleotide monomers, while RNA contains few thousands of nucleotide monomers. Nucleotide monomers consist of three components. Just now I said monosaccharide sugar, nitrogen containing base and phosphate group. So DNA on hydrolysis produces nucleotide as monomers and RNA on hydrolysis produces nucleotide as monomers. But DNA monomers are millions in number while RNA monomers are thousands in number. Nucleotide of DNA contains three components. Monosaccharide sugar, which is beta D2-deoxyribose sugar, is a pentose sugar, while RNA contains monosaccharide sugar, which is beta D-ribose sugar, and that is also a pentose sugar. Now you can just see in the picture that figure of ribose sugar, beta D ribose sugar. So ribose sugar is a pentose sugar, that means it is made up of 5 carbons. First carbon has OH on the top, H below. Second carbon has H on the top, OH below. Third carbon has H on the top, OH below. Fourth carbon forms hemiacetal and fifth carbon is CH2OH. So this is pentose sugar. And beta D, because it is beta D ribose sugar, so here OH is on the top, H is below. Now the sugar for DNA is beta D 2-deoxyribose sugar. So that means beta D ribose sugar is common in both. That means the basic sugar is pentose sugar made up of 5 carbons, and on the first carbon OH is on the top, H is below. Second carbon H and H. Third carbon is H on the top, OH below. And fourth carbon is forming hemiacetal. And fifth carbon is CH2OH. But here on the second carbon, what do you see? In the RNA, in the second carbon, H and OH were attached. But here on the second carbon, you see the two H's are attached. That means one O from the OH is removed in this DNA sugar. That is why the name is beta D2-deoxyribose sugar. So oxygen is lacking here. Oxygen is less here. So deoxyribose sugar. Then the bases present in DNA are of two types, purines and pyrimidines. Purines are adenine and guanine, while pyrimidines are thymine and cytosine. So the four bases present are adenine, guanine, thymine and cytosine. While in RNA, also the nitrogenous bases are of two types, but purines are adenine and guanine, and pyrimidines are uracil and cytosine. Okay, so here you see the bonds, type of bonds formed between A and T and G and C, and here A and U and G and C. So basically these nitrogenous bases pair and they form different pairs in the nucleic acid. The third thing present is phosphate group. So here you see this phosphate ester is present. So this is derived from phosphoric acid. What is phosphoric acid? H3PO4. So when H of OH in phosphoric acid is replaced by R, you get phosphatic ester. So these are the diesters, phosphate ester groups. Now see how they are linked. So this is the pentose sugar, let's say DNA nucleotide. It is made up of pentose sugar, deoxyribose, 2-deoxyribose sugar. So in the second, one oxygen is less than as compared in ribose sugar. Then on the first carbon, OH was on the top, H was below. So through OH, it links to nitrogenous base. So through first carbon in pentose sugar, it links to the nitrogen base. Nitrogenous bases are adenine, cytosine, guanine and thymine, and through fifth carbon it links to the phosphate group. So fifth carbon OH it links to the phosphate group. Okay, so that's how the bonds are formed. So water is given out here also, water is given out here also to form this bond. So this is DNA nucleotide. While this is RNA nucleotide is made up of pentose sugar, and on the first carbon it is linked to the nitrogenous base. Base can be adenine, guanine, cytosine or uracil, and on the fifth carbon it is linked to the phosphate group. So this is RNA nucleotide. So nucleotides are the organic molecules consist of nucleoside and a phosphate. So what is nucleoside? Nucleoside is made up of the sugar and the nitrogenous base, and this is linked to the phosphate group. So these are some of the questions. Define nucleic acid, write the representation of nucleic acid, distinguish between RNA, DNA. A very important question, write a short note on nucleotide, write a structure of nucleotide in DNA and RNA. So these are some of the questions. Thank you.